investigators at the Allen Brain Institute. His background is actually in applied mathematics and computer science, getting his PhD at MIT in this area. If this was a company, he might have been employee number 5 of the Allen Institute. He leads the data annotation and analysis effort and was the first author on the recent Nature paper on the brain atlas of the mouse. So I'm very happy that he was able to join us on short notice.
Thank you. It's a great pleasure to be here. Thanks, Chris, for this really exciting initiative, which seems to be getting off the ground. So what I'm going to talk about is going to be considerably more empirical in terms of the kind of data that we produce and some of the work that related to it. There's not much theory involved here, and because we haven't been integrated well with theory, and I think there's a lot of interesting opportunities potentially there. So, in any event, so I'm going to go very quickly through this. It'll be more like a kind of just a flash display of the picture. Let's see, what time should I stop at, Chris?
Let's try to aim to 20 after.
20 after, okay, yeah. I always like to say, if you've given enough talks, you can give any talk in any allotted amount of time. All you do is you just contract to it. So anyway, I'm going to talk about this. So the Allen Institute for Brain Science was started in 2001 by a very generous gift from its founder, Paul Allen. And some of the milestones here we have lots of scientific advisory boards for various kind of initiatives that we do. We believe in kind of polling the experts before we kind of get off the ground, moving in directions of things. People started to come in 2003, that's when I came. And we basically finished a major project, which was the mouse atlas, which I'll tell you about. And various other successes led to kind of a recommitment of funding from the founder. In fact, proposed building of this new building for us, which will be an actually permanent home just down the road. Right now we're kind of scattered in a bunch of kind of temporary places in 2015. This just sort of an overview of some of the kind of things. It doesn't have some of the absolute most recent things because they haven't really been per se milestones yet. But in any event, the first thing that we did was this large-scale in situ hybridization map of the mouse brain, the adult mouse brain. And basically what it meant was that this was sort of like the first thing of its kind. I think Mr. Allen had asked like the scientific leaders what would be something one could do on a large scale, which was repeatable, which would have large impact. And one of the things that came back was that to consider large-scale gene expression profiling in the mouse brain in a very high resolution sense. So we use this kind of in situ hybridization technology to basically kind of look at cellular level kind of expression of the brain and then sort of scan, in some sense, the entire brain and the axis of the entire genome. And so I'll tell you about that. Other things include, basically, for a while we didn't quite know where to go, and so we basically started exploring other axes there. For example, development, other species, macaque, etc. And then at some point we kind of tried to venture into the human realm, and that was, again, interesting. And there's an interesting trade-off with these kind of problems because you have, every time you open up a new axis, in some sense, you no longer can do everything. Even with the mouse adult, people wanted both coronal and sagittal and everything done in triplicate, and the costs in these kinds of large-scale things just multiply geometrically. And so you really have to make trade-offs and conciliations. This becomes even more so when you go into human. Just to give you a back of the envelope, to do the mouse atlas cost $44 million in terms of setting up all capital expenses and everything like this. Because of the essentially just 1,000 times volumetric expansion of the human cortex from the mouse, to do the same n=1 in situ hybridization atlas, or n=1.5, in a human would cost $100 billion. And there haven't been any takers on this funding opportunity. So in any event, if you go to our homepage, right here, you see something that kind of looks like this. Not the Institute homepage, but the application homepage, which is brainmap.org. And it basically shows you kind of a portfolio of various kind of atlases, things that people may or may not have had a chance to explore this. But The original mouse atlas is in the upper left-hand corner. Various other things follow down. This just shows a vignette here of gene expression. This is what characteristic in situ hybridization data looks like. You're looking at a part of the cerebellum there, and a gene which is kind of nicely expressing in the Purkinje cells, kind of aligning in the kind of that layer of a cerebellum. This whole thing is due to a very sort of large-scale, what Henry Markram had called the industrialization of neuroscience, basically using as much robotics as possible, as much automation as possible, a lot of automatic things in terms of the computer analysis, such as image registration to where possible. And it requires, you can't completely automate these things, but you can do them in high throughput, provided you have the right kind of injection of checkpoints and places for correction. And that's a kind of a key thing. But basically, we kind of explored basically about 24,000 transcripts in the mouse brain, adult C57 black 6, and everyone was cut sagittally, the long way for starters, and then about a quarter to a third of those genes were also done in coronal. People generally like coronal better because you see smaller amount in more detail. Sagittal is better sometimes for more pan aspect of the brain. This just shows about the slides, various kind of these TCAN robots which were capable of running this in situ representation, various kind of repeated tests to show that the same signal was coming. This is the kind of thing that you would see. We're looking at right here a gene, monocytase 1A, basically, just showing the kind of thing that the complexity of the gene expression, in essence, is that you see that this is a gene, and as to what does it mean for an in situ hybridization image to actually be expressing. That in and of itself is also kind of it's a bit of a debatable kind of thing, because the process is semi-quantitative in some sense. Particularly colorimetric in situ hybridization, which is even less quantitative than dark field, radioactive dark field. But you can see in this gene that it's expressing this pointer. Oh, thank you. So, yeah, where's the right one? Anyway, expressing here clearly in the striatum, places of the olfactory bulb, looks like layer 5 in the cortex, going across here. So this would be the cortex of the mouse with layers here, completely lissencephalic, not at all, not sort of folded at all like higher mammals. And there's the cerebellum over there. Basically, in this kind of data, because of this, what you get that you don't get out of something like, say, microarray or RNA-seq, is that you get spatial context, right? You can't, everything that we're seeing here, this is cellular resolution. We do image these at 10x, sometimes 20x. But what more importantly is you see the relative nature of what's expressed compared to its environment. And that's very, very important for understanding kind of relationship and structural information. Here I'm just showing a bunch of markers. Interesting, when we started this project, no one really had any idea to what extent genes would be, what the nature of gene expression was in the brain as a whole. There were many, many isolated studies, many things done in situ hybridization, lots of microarray work, but nothing really, no sort of overall gestalt of what is gene expression in the brain? How many marker genes are there? We perhaps thought that every region of the brain had a definitive gene that would demarcate it because it had to be defined, or what was the combinatorics of it, etc. And so, in many cases, this is major cell types are delineated, like we're looking here in the hippocampus, dentate gyrus, various things, CA3. But in certain areas of the brain, it's much more subtle, much less kind of straightforward. To do this and make sense of this, you need a frame of reference, like a reference space. And so here, what we're showing is that this was a reference atlas, analogous to the Paxinos atlas for the mouse, Paxinos franklin, which was drawn by Hongwei Dong, who was one of the original people of the institute, but now it's at UCLA. And he basically went through, this has been refined since his original drawings by new neuroanatomists, but basically this is just showing a coronal section, the Nissl staining a effectively most of the neurons on the left, and then a kind of its delineation on the right side of the brain, and color-coded. This color coding was not just for a glamour, but actually color coding the ontology and having these things, the images sort of color coded, where every distinct region down to the lowest level has some different RGB value, is used critically in a lot of our machine processing algorithms, because you can recognize in constant time exactly what pixel, where it should be, right? And so in terms of like assigning expression to regions, this became an important thing. There's basically a very large-scale kind of informatics pipeline behind this that basically starts with the reference atlas, assembles that into a 3D space, takes this in situ hybridization data, essentially builds a 3D prototype brain from that, and then registers it using image processing algorithms, a combination of information and other based algorithms to basically deform that into the 3D geometry of the brain from the atlas. And then we grid it. At first we thought, well, maybe you don't need to grid it. All we do is you have an atlas, you know what's expressed where. Perhaps we can just pool expression at the ventral tegmental nucleus, or pool it at lower part than some striosome of the striatum. And in fact, but what we thought kind of closer toward as we got it, well, we'll put this grid on the data anyway. We basically put a 100 micron grid across the entire brain. And then basically only pool gene expression by these grid statistics, because then we can always unionize it up to structural level. This turned out to be a very, very good idea. And the reason is largely because essentially the combinatorics of gene expression in the brain. There are, if you decide, one of the things that people said in the beginning, they thought, well, you're going to need search algorithms. People are going to want to know, like, what's expressed in the arcuate nucleus, and you're going to want to return genes that express there. And it turns out that that's not as easily defined as it sounds. And the problem is it's not, it's too general. There are too many genes expressing too many places, in too many combinations, and too few high information content markers to really make such a search really effective. And so you have to use other kinds of means. We basically, you need to use more kind of correlation-based things, information-based searches, and I'll tell you a little bit about those in a minute. But one of the consequences of this, we can take Vanadase 1A and basically now visualize its expression in 3D here. And now these little spheroids here are not, they're no longer cells, but they're the pooling of the expression to this 100 micron grid. So you're basically just seeing a realization. It becomes kind of a gestalt for the gene kind of experience. And this is clearly the hippocampus with its high kind of expression there. Standardization is really important for sharing and for kind of information, kind of communication and stuff like that. And so people want, a better view of neuroscience is not sort of one ontology or one standard, because everyone, that people will, they have these meetings, for example, anatomists will have these meetings, and other people, computer scientists, and we have the very noble goal of discussing ontologies and locking down ontologies. And it's a bit of an endless battle. And part of the problem is that not only because people see the world sort of differently, but that people have, for historical reasons, they're forced into using certain kind of paradigms for various other technical reasons. And so a better model is to have various skins, of course, for the thing, even ontologies, regions of the brain, etc., that you can say, well, according to this theory, how would I look at my data set? And sort of this interchangeability is very important. And one initiative in this that I have been involved with is this INCF group, which is the International Neuroinformatics Coordinating Facility in Stockholm, is very interested in kind of building standard spaces for things, in particular for this, the mouse brain atlas. And so we call this place Waxholm Space. It's kind of a kooky name, but it's named after the town where a meeting was held. But this is basically a space to which others are able to map into, so we can provide data interchange to talk about the same areas of the brain at the same time. So going back to this gene here, this monosidase, taking a little bit closer look here, you can see I've zoomed in a little bit, and I'm showing you now the sort of clear expression in the striatum. Not as clear what's going on other places. For example, this little cell group over here, probably, excuse my neuroanatomy for those who really know, but probably in the midbrain over here, or maybe even in the pons, but is that expressed, or is it just a high-density cell group? It's not completely clear in actuality. And I say that based on having looked at a lot of images, not looking at this image in particular. But the issue is to how to define search, how to access this data and manage it. And so one of the things that we developed as a way is this kind of just correlation-based search, where you basically now, we're going to basically just discretize that using that big grid of data. We're essentially going to essentially look at that grid either as a linear array of voxels or as a voxel with spatial relationships. You can do it either way. And essentially say, find genes like me, find genes that have a high correlation or high information content with the expression of what I have in my seed and others in the data set across all 20,000. And this works pretty well. You can, for example, I'm doing ETV1 here in layer 5, or this nephroblastoma in the CA1 of the hippocampus, and I'm able to return genes. And so you can do this from the website. Actually, the interface has changed a little bit. This is a slightly older picture because I couldn't get it last night. When I wanted to put a picture of this in there. But it's essentially the same thing. You can choose a gene, choose a region, and return correlates. And so here I've done it for the sort of tryptophan hydroxylase, a gene which is basically, let's see, right here on the top, basically expressing very, very distinctly and almost exclusively kind of in midbrain, hindbrain area. It's a protein controlling the rate enzyme, rate limiting step of serotonin production, as we know. But what I can do is I do this correlative search, and I can return things that, whereas they don't look exactly like that, in the grand scheme of the expression across all genes in the brain, these are very, very highly correlated. Okay? And then, and in fact, they share a lot of the same functions involved in kind of satiety, potentially drug-related activity, et cetera, like that. There are other approaches that one can take. In some sense, there's kind of I like to say like a three-pronged approach one can take to this kind of management of the data. Is that one is, well, you can simply do search by structure. You give me a structure, I tell you what I know. Or search for a gene itself. You may have most of the users simply have genes that they're interested in. I'm interested in ETV1. Tell me what you can tell me about that. And I've talked to you about this thing I call neuroblast, which is this correlative search. But one can take a slightly orthogonal kind of direction here, which is basically saying, I can think of it, it's almost dual to the idea of talking about this kind of correlative search by voxel, and that's to look at a voxel now and say, I'm going to give you the vector of all genes expressing at that voxel, everything I know about that spatial location, and now I want you to tell me how does that relate to other spatial locations with respect to the gene set you've given me. For example, I could consider ion channels, and I could say, I'm going to look at a voxel that's in somewhere in the cortex for ion channels, and I want you to tell me in the class of ion channels, how are other voxels related to that in terms of its expression profile? And this that we call it a GIA. People like that name, neuroblast, but they didn't like the name of GIA. They thought it sounded like a car. But it stands for Anatomic Gene Expression Atlas. And it basically is, what you can do is you can navigate a reference space in the reference atlas and basically place a cursor someplace of interest and see how is the expression values at that location correlated with other parts of the brain. And you can then, and in fact, it would be more toy-like, except that you can use that as a template to return genes which are highly representative of that pattern. Okay? So these are the kind of tools that we built for this sort of thing. This is just showing its use in identifying, sort of shedding some light. This is a paper that we published in Nature Neuroscience some years ago about studying kind of gradients in the striatum and whether gene expression can shed any light on the kind of classical dogma of dorsal versus ventral kind of partitioning in the striatum. And you see that, in fact, there was, consistent with people who had hypothesized more recently of a kind of the ventral lateral kind of medial axis, that in fact you can see that this was borne out by the gene expression through some 3D modeling, and then we were able to go back and find genes that kind of bore that out. And others have done the same at campus. This is another publication by I'm going to blast through this stuff a little bit more here, just to make sure I stay on topic. on time, but you can extend this. We extend this into the developing mouse by doing 7 different time points, from P11.5 all the way up through the adult, and then an aging mouse to a 2-year-old mouse. And so by this point, the ISH process, we had really honed it in the sense that some of the earlier genes in the first part of the adult atlas were probably of lesser quality than as we went along. We were kind of refining the process a little bit. And, but by the time we started this project, we had really kind of got it down, the automation of it. And so the data of these basically 2,400 genes sort of now again, like I said, every time you add a new dimension, you sometimes have to cut back on your sampling. It's a problem of wanting to sample a larger and larger space. You can't take as much data, unfortunately. And so you have to you want to do more of a directed kind of a search, almost like an importance sampling, if you will, of sorts. And so what we do is we basically looked at genes that were somewhat driven by what we found to be interesting in the adult, as well as advice from the kind of neurodevelopmental literature and experts. And we kind of compiled about 2,400 genes germane to development, and kind of partnered with people to collaborate to do new Luis Puelles, a well-known developmental neuroanatomist from Murcia, Spain, and basically to develop a series of new reference atlases for each stage. And then we kind of generalized all those tools that we built, all those correlative tools to the developing animal too. And you can find all those online. So they're showing that reference atlases, different things, 3D models, etc. Search now takes an interesting thing. Now you have a spatial and a temporal direction, and you can kind of control that axis. You can search across different things. You can study the progression of data across time, and all those tools sort of generalize. This is a paper one of those papers that we've been trying to write up, but somehow we never seem to finish, trying to talk about this particular aspect of the work. This would be a figure if the paper ever got published, but we're still in the works. So, other things is connectivity. So we know, in recent years, in the last few years, there's been a really big interest in terms of from the NIH and some other funding agencies in understanding the sort of mesoconnectivity of the brain, of the rodent brain. It's sort of what's projecting to what. This is not so much of it's more of a projection atlas than it is a connection atlas, because we're not talking about actual contact, synaptic contact, or really modeling or studying that, but really just getting a sense on what nuclei are projecting to what nuclei. And so to do this, there's at least three efforts in the world going on about this now. Some use more classical kind of virus-based kind of methods to basically study it. The approach we're taking is by basically, we did some of that, but we're also using kind of transgenics. We have a big transgenics program that basically identifies markers and then basically uses kind of Cree reporter lines to basically engineer mice that definitively will express for various genes and highlight their kind of projection and kind of connection. And so we invested in all these sort of this interesting system called TissueSite, which is basically, it in some sense solves some of the problems that we had when we did the adult atlas with regards to the informatics processing. Because these machines, what they do is that you can put a brain in there, and it's a small company. It was one of those companies that is from Boston, a small company from Boston called InfoSite, and they sell you the thing, but it's still under development, and you kind of have to help them along, and eventually you get it right. And now we have about 6 of these machines, and basically what you do is you can put some piece of brain. You can't put a human brain in there, but you can put a mouse or even a macaque brain in there, and it basically does essentially block face imaging while it performs a 2-photon microscopy. Okay, so you can basically it gives you a block face image, so you know exactly what plane was taken, and then it does basically the 2-photon, and then it moves on to the next section. And now what's nice about this is that this solves the problem of registration, essentially, because since you have the block face image, it's very easy to associate tissue sections with a pristine prior to cut image. Okay? And this really enhances, it's not, we didn't choose this so much because it was absolutely necessary for the connectivity atlas, but we chose it because it was available and we could actually really make some progress. It just shows sort of various kind of transgenic kind of reporting lines here of different areas of the brain, different parts of the cortex being expressed. You can really control what's going what. And so we're building up a big atlas of this. It's slated to be completed probably in the next year or so. But it leads to a lot of interesting data. You know, this is just showing kind of a connectivity matrix here of what's projecting to what, a first pass based on some of the data that we have. It's called modeling. So in the last segment here, I'll talk a little bit about our human effort. So you move into the human. As I said, you move into the human realm, and all of a sudden now you have to cut back even more. Doing in situ hybridization on one specimen or one and a little more specimens for the whole brain, as I said, it would be prohibitively expensive. And the question is, whose brain would you choose and how valuable would it be? It's not really likely to be that interesting necessarily. You know, you can't really confirm it. You don't know where there's more diversity, and there are no wild-type humans, at least politically maybe, but not scientifically. And so basically, the approach we took was that we essentially scanned the brain in a prototype kind of way using microarrays. We started this before RNA-seq was even really feasible. They were just kind of coming online. We're now, for the later part of this project, we're actually doing some RNA-seq now. But the original was basically, for each brain, we sampled 1,000 locations in the brain and ran a custom 64-array chip at these 1,000 locations. And then we also did in situ hybridization of specialized areas that were more interesting. This just talks about the whole workflow here, how things are sampled. I won't go into the details. I just want to share with you some of the, there's a lot of technology involved in cutting and dealing and managing these large brain things. This just shows you the two types of sampling they did. Some things were cut out by dissection. Some of it was done by laser capture microdissection to get fine structure when we needed to do that. In the end, a lot of the same tools were generalizable. We could build inflated base maps of kind of gene expression. This is showing two brains, an inflated representation of the MET proto-oncogene. A very interesting gene. It's perhaps one of the most differentially expressed genes in the whole human brain. Has a very, very strong kind of posterior-anterior gradient in it, even more so than it seems to be here. But it's been implicated in various kind of disorders of autism or other things potentially. Quite interesting. But the kind of thing that you can do with this data set is, for example, here's a picture that out of this recent paper that Chris mentioned, is that there's a type of analysis here where I'm just showing, I'm going to look at the differential expression of the human brain. And this is basically formed by pooling 3 subjects. And I'm basically, in other words, what I'm doing here is let me tell you what it is first, is that it's basically about 150 by 150 structures. So the samples are pooled up to a smaller structural level. And then we basically kind of compute gene expression, and we can create the differential expression subject to hypothesis testing, correction, and everything between these 2 regions. So basically, what this means is that a dark region here means not much differential expression between the 2 regions admitted, whereas something brighter means quite a bit more. And so let's just look at it a little more carefully. This whole upper area up here, in the upper left, is the cortex. Is that it's showing a very homogeneous cortex, not much consistent with the kind of the common architecture theme that's been talked about here today. But basically, throughout these, say, almost 300 regions of the cortex, basically comparatively small differential expression between these regions. Number of genes. This is counting the number of genes that are differentially expressing, right? Crossing some threshold subject to correction or thing. Except a few places, like this one place right here, this band. This band right here, which is actually a V1 in the visual cortex. And there are a lot of genes that are very differentiating between V1, which is quite interesting. And other places here, very temporal pole. But this is all subcortex. A lot of nuclear kind of differentiation separating kind of gene expression subcortically. And then you have right here in the middle, you have sort of a striatum with all its kind of, which also has a strong white matter component. which differentiates itself from the neocortex considerably. And a lot of interesting features are kind of just sort of derivable from this gestalt kind of. More or less lastly, except for a few final words, I'll just show tell you another result from that. Is that, so even though there is, in some absolute terms, little to distinguish itself from gene expression, there are still slight changes that are interesting and have meaning. And if you look at that more carefully, you restrict yourself to the cortex, you can do this interesting experiment. Is that here's your topography of the cortex with various regions labeled: frontal pole, parietal, superior medial gyrus, inferior orbital gyrus. They fall into the basic lobes of the brain that we know. You can, what we can do is we can pool data to these small gyri in the same way we did in the previous figure, and we can consider the gene expression relationships. And to do that, I'll use a standard kind of multivariate technique called multidimensional scaling, right? Which probably a lot of people will be familiar with that in an audience like this. But basically of course, multidimensional scaling is basically just a close relative of principal components analysis, right? It's saying, how would I represent my data in a lower dimensional space if I chose and fixed the number of dimensions, right? And it turns out that the optimal is very easy to show is for you to choose those directions of the first k principal components if you're looking for a k-dimensional space. Now, so this is a realization in 2D here, and I see this interesting characteristic. So what I'm looking at here is how would these gyri be placed, and how would the samples be placed if I let gene expression alone dictate their position? In other words, how much does gene expression tell me about their position? And basically what this is showing, first of all, just conceptually or qualitatively, is that the lobes are more or less coming out into their right locations. And in fact, the relative position of gyri within the lobes. And so how do I choose a distance metric here? Well, I guess that's the main point I haven't told you. Distance is chosen simply by differentiation between the two regions. And to account for sort of like not oversampling certain ones which may have more priority, I allow every pair to have only one vote. For every pair of regions get to say, give me your best gene that says I'm different than you. And that's the gene set that goes into this and produces this result. And it basically, if you do this in 3 dimensions, which you can't, I'm not displaying here, if you do this in 3 dimensions, you can actually measure how accurately you really recover the kind of the cortical architecture, and you get about 30%. So you can, in some sense, imagine that gene expression determines at least a modest amount of the architecture. So just to finish, and my time is up, is that this is by no means in any way complete, is that a lot more refinements and results are necessary. Our delineation of the primary visual cortex in the mouse is just a blob, yet we know through the work of sort of Wang and Burkhalter that there's through projection studies that there's a lot of interesting architecture to about perhaps 10 regions in the visual cortex of interest. And so in a new initiative that's kind of come about, that in fact Dr. Tononi is involved in, is this thing we call MindScope, which is a much more kind of pan effort to try to understand in more detail the mouse kind of visual cortex. And basically it is to combine multiple data modalities from physiology, gene expression, connectivity, etc., to try to understand from input to coding, to neural coding, what kind of is the full kind of spectrum of the architecture. And so this I don't have time to talk about that, and since it's just getting off the ground, it's not really the best thing to kind of for a talk perhaps. But essentially there's different teams that are involved, a cell type team which will be involved in sort of studying cell characterization. It's always a sticky topic. What does it mean to characterize cell type? But the idea is that you try to do enough to use it as a differentiating mechanism as opposed to completeness. A modeling analysis and theory team whose existence and its kind of the quality of people we're getting is much benefited by Christoph Koch, who's joined us at the Institute, and also a neural coding to do with sort of understanding circuit level dynamics and circuit physiology. So basically, this is of course the work of it's a collaborative work of an enormous number of people internally and externally. And I hope I've given you a reasonable survey of just the